In a muscle cell, the contractile proteins are arranged in long sticks called myofibrils. Looking at a single myofibril, you can see a repeating pattern of light and dark lines. Each segment from the middle of one light area to the middle of the next light area represents a single sarcomere. When each sarcomere contracts, you can see the whole myofibril shorten like a slinky or an accordion. Let's look at an individual sarcomere. It goes from this zigzag or Z line to this Z line. In a more detailed view, we can see the contractile proteins involved in muscle shortening. The thick filament, shown in blue, is called myosin, and the thin filament in orange and yellow is called actin. Myosin is made of many individual molecules. Each is made up of a long arm and two globular heads. When activated, each myosin molecule can lift, then pivot or crank. This is called the power stroke. Now the myosin is stuck in the up and cranked position and needs to be reset. The energy molecule ATP comes in and binds to the myosin head, and the energy it imparts causes the myosin molecule to be reactivated or reset back down along the myosin thick filament. When ATP is hydrolyzed, the crossbridge is energized and ready to go again. Like setting a mousetrap, the energy is imparted into the crossbridge head in the resetting. Let's look at the orange and yellow actin molecule. See that actin is attached to the Z lines? Looking more closely, we can see the actin beads strung and twisted together. The yellow region represents the active sites where myosin molecules attach. The regulatory protein tropomyosin looks like a long green rope that covers all of the yellow sites on actin. When the yellow active sites are covered up, the muscle is at rest. Troponin is a regulatory protein that's spaced out across tropomyosin. We can see it as little brown dots on top of the tropomyosin rope. When calcium is released inside a muscle cell, it binds to troponin. The calcium-troponin combination will cause the tropomyosin to move over. This exposes the yellow active sites on actin, as we can see here. It is at this point that the myosin molecules can lift and bind to these active sites. Now we can see them together. Let's go through this sequence step by step. Once the muscle and inner myofilaments receive the signal, being calcium, to bind to troponin, this allows tropomyosin to expose or uncover these yellow active sites on actin. When the active sites on actin are exposed, each myosin molecule will lift and bind to that active site. Once the myosin molecule connects to the active site on actin, it forms a crossbridge. The myosin crossbridge will pivot and move actin, the entire actin strand, over. This action is called the power stroke, and this is what causes the Z lines to be pulled inward during a muscle contraction. After the crossbridge head has pivoted and pulled on actin, it can only be released and detached when ATP binds to the head. It is the binding of ATP and the hydrolysis of that ATP molecule that allows the myosin molecule to lay back down and the head portion be reset. At this point, ATP has split into ADP and inorganic phosphate. Now it is energized and ready to go again and repeat the cycle. We can see this cycle will continue as long as the active sites on actin are exposed and there is sufficient ATP. As a whole sarcomere, we can see this pulling the Z lines inward. Let's look at the effect of a sarcomere contraction along a myofibril. When each sarcomere contracts, you can see the whole myofibril shorten like a slinky or accordion.